Everybody say hi.
Hi.
They all waved. All right. So as I say, you may know, I hate the little homework assignment things or weekly quizzes. If I was to give you a weekly quiz, it would be this. What are the 4 macromolecules? What are the monomers that make them up? And give me 4 interesting facts about each one. So that's what we've been covering so far. The idea about the different macromolecules, carbohydrates, lipids, proteins, and nucleic acids. And one of the key points here has been the idea about how we've got different bond strengths involved in these macromolecules, and the covalent bonds that put these things together are very tough. You can't break them unless we either do enzymatic wizardry, which we're about to get into, or you heat them really, really hot. But the weak bonds, them weak-ass bonds, that's what's giving a lot of different things structure or making the carbohydrates gel. Phospholipids, whatever. Polar, non-polar interactions going on there. But certainly with proteins and with nucleic acids, we can denature them. Like when we denature proteins, we add a little bit of heat, and primarily we're breaking the hydrogen bonds, but then we can add salt, we can change the pH, we can do a whole range of things and change their secondary and tertiary structure, make them non-functional. With nucleic acids, it's more, if we add heat, boil it, we can take the double-stranded molecules and break the hydrogen bonds between bases and generate single-stranded DNA or single-stranded RNA. DNA is going to try to hybridize back to its base pair complement, and it does a fairly good job of that. So DNA will renature pretty well. RNA has the ability to fold up, sometimes depending on the sequence, to make fairly complex intramolecular base pair structures. And there you go. So again, another point would be that carbohydrates don't have an end to them. That is, there's not like a 5' end or an amino terminus. They are pretty much homogeneously negative charged. Lipids don't really fit into that sort of model, so we have the whole lipid story. But when we start talking about proteins, we've got the amino terminus and the carboxy terminus. You can only add things to the carboxy end of a growing protein chain. When we're talking about nucleic acids, we have the 5' end with the 5' phosphate and the 3' hydroxyl, and we only add to a free 3' hydroxyl. And these are just fundamentals to keep in mind because it helps us to think about the orientation when we start thinking about polymerizing RNA from a DNA template, when we're making an RNA sequence, and then also when we start making amino acid polymers, proteins, from an RNA sequence. Keep that in mind, and we'll be doing good. So the next step in our discussion is based on thermodynamics, because what we're going to be talking about pretty much from here on out is how different processes happen. And everything from how we can polymerize nucleic acids to how we move salt in and out of the cell is going to depend on proteins and their ability to do things. And fairly quickly we'll get to the spot where we're going, okay, the sodium-potassium ATPase pumps out 3 sodiums and pumps in 2 potassiums. And the actual mechanism of what the protein's doing is kind of like, it just does its thing. What we're going to talk about now is some of the fundamental mechanisms about how proteins actually can do stuff. And this gets into a little bit of basic thermodynamics in terms of how we can begin thinking about these kinds of chemical reactions and how we can begin thinking about proteins' involvement in it. So what we're going to do is start out with build sort of a chart comparing different kinds of metabolic processes. But first, we have to start out with the fundamental laws of thermodynamics, which really, this whole thing is driven on thermodynamics. So we've got a couple of laws of thermodynamics that you may be familiar with. So these are laws. We do not break the thermodynamic laws. It's bad manners. the first law, very critical, and that is energy cannot be created or destroyed. And this is the first law of thermodynamics, but the truth is, for what we do, it's neither here nor there. I'll take it as on faith that, yeah, you can't create energy, whatever. We can transfer energy in different forms. We have what you think of like kinetic energy versus potential energy. You can heat stuff up. You can do some work with that heat. So for what we're going to be talking about, while that's the first law, fine. The second law is really a bit more important to our discussion, and that is the following: that all systems, and this is life, the universe, and everything, all systems tend towards what the thermodynamicists would say is entropy. We can think of this as disorganization. And this is an absolute fundamental truism here, because what we got is the idea that systems are not able to self-organize without energy input anyway. And if you leave things alone, they fall apart. So the situation is going to get worse unless we do something about it. What it means is we can predict which way a chemical reaction is going to go unless we do some kind of enzymatic wizardry or put some energy into it. And that is, it's going to fall apart. Another way of thinking about this is, left alone, big molecules are going to begin to break down and decompose to smaller molecules. So, this is a driving factor in how we think about chemical reactions, because from here, we got these two terms we can use. If you're following the second law, you have a reaction that is said to be spontaneous. So a spontaneous reaction is a reaction that is tending towards disorganization. Another way of thinking about this is we're going from the more complex to the simpler. So we have really complicated systems. You leave them alone, they're going to start to fall apart, they're going to degrade, the molecules will begin to decompose, and you go from a more complicated system to a simpler system, more energy in the system to less energy in the system. That we can guarantee. And a spontaneous reaction then is going to follow the second law. Now, the other side of this, and I'll go over here, is what we would think of as a, for lack of a better term, it is a non-spontaneous reaction. And this is where we're going from simpler molecules and generating more complex molecules. This isn't going to happen unless we add energy into the system. And we're going to have some fairly clever systems in place to direct this energy to actually do useful work. But there you go. Pretty much what we have described here is the balance between the different kinds of reactions in a living system. You've got reactions that are breaking things down and generating energy, which I guess I should say that. So more complex to simpler. Energy is released. Consequently, breaking stuff apart releases energy. Building stuff up takes energy. If you match these things together just exactly, you're golden, you're set. So we can build a fire and get lots of heat out of it. You could then take that heat and turn it into a steam engine and do some kind of work with it. Metabolically, we've got things a little bit more complicated, which is what this entire course is all about. But what we're now going to do is set up a kind of a ping pong comparison chart between these two different types of metabolic pathways, two different types of metabolism. And that's our next big chart. Okay. So one of the things that we have to keep in mind is that we use this word spontaneous, but it's not the way we would think of spontaneous in a standard kind of conversation of something that's going to happen like that. Spontaneous, like spontaneous combustion, which doesn't really happen very often. A spontaneous reaction doesn't necessarily happen fast. It is just we know that's the direction it's going to go. So we got to be thoughtful about that term. But what we got here then? I'm going to close the door. Someone's laughing uproariously. What we got then is coffee. Oh, mother's milk. So, we can break down the biochemical reactions in life into two different categories. What we've got is on this side is anabolic reactions, and on this side we have catabolic reactions. And we can pretty much view these as two sides of the same life coin. We're going to ping pong back and forth between these two. If one can, the other can't. If one does, the other doesn't. All right. So to define them, first off, anabolic reactions. These are taking simpler molecules and generating these complex polymers we've talked about. Catabolic is exactly the opposite. We're taking complex molecules and breaking them down into simpler molecules. So, just a real quick example, anabolic reactions are all those reactions you do when you're building muscle, when you're making new polymers, you're generating new molecules of nucleic acid, you're making sugars, all this stuff. Catabolic reactions, that's what you're doing when you're breaking stuff down to eat, when you're digesting stuff. So we got these two different kinds of reactions, and just a sort of a way to keep in mind or to remember them is that people will take anabolic steroids to roid up to try and favor this side of metabolism, and nobody takes catabolic steroids. Catabolic steroids, that would increase breaking stuff down. I got enough catabolism in my life, and catabolic metabolism is stuff like drinking, smoking, staying out too late, busting yourself down. That's catabolic. We don't need to take things that will push that side. I do that fine enough on my own. What we're always trying to do is sort of push over to this side. Now, it is pretty much a perfect balance. I mean, unless you are like starving, then you got some troubles because you are breaking things down just to try and maintain energy. Anyway, if things are cool, this is what we got, a perfect balance between these two different systems. Now, as we go through this, given this basic definition, a few things fall out. First off, anabolic reactions are not going to be spontaneous. These are non-spontaneous, as we just discussed. Spontaneous, and meaning that we need energy input. And the exact opposite's over here. With catabolic reactions, these are the way things are going to go thermodynamically. So we would say these are spontaneous. Now, they're not going to happen, you're not going to fall apart all that fast. But that is the way you should bet because, things are going to go that way. And these are going to be reactions that will release energy. Okay, so as long as you balance those two things, you're good. I mean, when you think about this from a diet standpoint, we're talking about calories in versus calories expended. Many of you may have like your little watch Fitbit that measures the calories that you've expended during the day, and it does it by measuring your heart rate and probably looking at like galvanic skin response. How accurate is it? Who knows? Those of you that are doing is anybody swimming anymore? But I know often hear about swimmers are consuming massive amounts of calories because you swim for a couple hours and you are burning through a ton of calories. As long as you maintain the energy in versus the energy out, your weight stays the same. If you start shifting one side or the other, you start eating more stuff, releasing more energy, but not doing anything, we gain weight. If we start doing more stuff and not putting as much energy into the system, you lose weight. And for the most part, if you're kind of keeping track of what you're eating and how much exercise you're doing, but you just stay right there in the middle, which is kind of a nice thing to do. I guess it depends on where you are. All right. Now, a couple of other things, sort of factoids from this that are useful to think about. So we got energy being put in and the energy being released. Additionally, these kinds of anabolic reactions, they are often reduction reactions. And these are typically oxidation reactions. And if you think back in your chemistry, or not, we can define oxidation reduction pretty easily, and it is this. Oxidation is loss of electrons, and reduction is gain of electrons. And there are a number of clever mnemonics for this. My go-to is oil rig. Oxidation is loss, reduction is gain. Remember that I did my PhD or my bachelor's in all my chemistry is at Texas A&M University and Texas with lots of oil. This made sense. You can also say loss of electrons is oxidation, and that is Leo. Gain of electrons is reduction, and that is Ger. And you say, Leo the lion says Ger, except that's not how you spell Ger, and so I can't do that one. Sorry. Oxidation is loss, reduction is gain. I stand by that. All right, so what this then means is that when we're carrying out catabolic reactions, we're going to be dumping electrons out. We're going to be liberating the electrons. When we're carrying out anabolic reactions, very often, we're going to, particularly when we're building up molecules, we're going to need a source of electrons to go into those new covalent bonds. So as long as we balance these two sides of the metabolic coin, we're good. If one side is starting to kind of get ahead of the other, there can be some complications. So this is how we think about metabolism. Now, we still have a couple of issues, though. We start thinking about living cells and living creatures and living organisms, about how this is going to work. And it comes from this. So we're now going to start talking a little bit about how we can carry these reactions out in a living system. So here's something to think about. We want to break down a macromolecule, like, just for the sake of example, we're going to break down a carbohydrate polymer to generate some energy. And you could do this, thinking about like a log or very finely powdered sawdust, different things. We can make fire. Yes, let's make some fire. And so we'll get some heat out and do some work with that energy. That's a good way of thinking about this. So what we're going to talk about now is some enzymatic or defined enzymes and things. And it fits, the story is easier to see if we think about catabolic reactions. So we're going to take that first. So if we think about a chemical reaction in which we're breaking stuff down, we can plot it like this. We're on the y-axis. We're going to put in relative energy. And it can be high or it can be low. On the x-axis, we're going to have reaction progress. So, just as an example of thinking about wood and fire, we got a chunk of wood up here, or a bottle of gasoline, whatever. So let's put a piece of wood up here. Just to be complete, we're going to add oxygen. When we start getting into the nuts and bolts of metabolic pathways, we'll see how critical oxygen is. I'll just give you, a heads up. You can't set fire to a piece of wood in a non-oxygen containing atmosphere. Don't try it, it won't work. So if you don't have any oxygen, you ain't going to get no fire. Because fire is essentially very rapid oxidation. You need a place for the electrons to go. They're going to go onto oxygen. And what we end up doing is dumping electrons onto oxygen. And we generate carbon dioxide and water. So this is the end result of the complete breakdown of a chunk of wood in the presence of oxygen. Fireplace. So, natural gas for sure is burning really, really cleanly. At least you depend on that. But even with a fireplace, if you've got like a glass screen in front of your fireplace, you actually can see condensation because you are generating water in the form of water vapor that condenses when you're breaking down wood, when you're setting on fire. And you're, of course, liberating carbon dioxide. Another way of thinking about these things is stuff up here is not going to be real good fire putter outers. Stuff down here is really good fire putter outers. So we don't typically have a fire extinguisher that is spraying liquid oxygen. That would not be a good fire extinguisher. Or a fire extinguisher that would spray a fine mist of gasoline. No, don't use those things. CO2 and water, yeah, they work really well. What they're doing is able to contain the fire. Carbon dioxide is going to be starving the fire of oxygen. It's heavier than oxygen. They also don't have a lot of energy in them, so you're safe using these as fire extinguishers. So now, the energy out is basically that. And so what we've got then is heat, at least in the case of our fire. This is the energy released. Okay, so now, with this level of thermodynamics in mind, and this level of sophistication, I say I'm going to build a fire. So I take logs, set them up, pour a little bit of oxygen onto it just to kind of get things going, and then stand back. What did I forget? What's wrong here? You need a match. Oops. Crap. All right. So let's throw in a match. The match actually is going to look like putting energy into the system. So that is what we have to have a little bit of extra energy to get this thing to go. This is known as the energy of activation, also known as the activation energy. So, here is the rub. It's a spontaneous reaction. I was told it's spontaneous, by everything we've said here. I should be able to set up a log, pour oxygen on it, and stand back, Damon, because I know thermodynamically it's going to break apart. Give me some heat. The problem here is you can wait like a couple of thousand years. I don't know how long you have to wait. But that is the way it's going to go. I mean, it is eventually going to decompose. Wood, as it turns out, is pretty tough. You're probably going to have to wait quite a while. And the energy will slowly come out. But what you know is not going to happen is that wood is going to self-assemble into a little house with little people walking in and out of it. So we know it's going to fall apart. It's just going to take a really, really, really long time for it to decompose. But if I add a little bit of energy of activation, I can get things going a lot faster. Okay. So the energy of activation and the activation energy, it's not that much. I mean, a match, at least from what I read, is about, burning at 700, 800 degrees for a very brief moment. And if you've ever been in, scouting, you can build a fire and you can get it to ignite with a single spark, kind of flint and steel, or a whole different way of, lots of different ways of setting fire to stuff, not using matches. But you got to really set it up very carefully, and you can get that little teeny single spark, poof, to go. And so the energy of activation really, it can be intense, but not very long and not very much. We're good. The problem is, now, I can't digest wood at all, but if we want to just, rather than saying wood, let's just have a 5-pound bag of flour. And I'll tell you that a 5-pound bag of flour, if you puff that into the air and throw a match into it, it'll go off really, really well. You can make yourself a really big explosion that way. The activation energy required is pretty darn high. I don't have the ability to generate that kind of heat. Be a cool superhero trick if you could, but as we've already discussed, if I had pretty much in any part of my body the ability to generate, 400 degrees, 500 degrees, enough to set this thing on fire, I'm going to denature proteins all over the place. I don't have the way to, even in an intense way, to generate that kind of energy. We can discuss fire-breathing dragons. I have some theories about that, but that's for a different discussion. That's for a different class. I am, just by the way, fascinated by the molecular genetics of dragons, but we'll talk about that later. So, I need a way that I can use the energy that I can generate. So, on a good day, I can pull about 100 degrees centigrade, or 100 degrees Fahrenheit, 37 degrees centigrade, yeah, God, I'm not the Human Torch. But I can pull 100 degrees. What's 100 degrees going to do for me? Not jack, sorry, but that's the best I can do. I cannot generate, running a fever, I might be able to do 105. When you're a little kid, you might be able to do, 105, 106. But as you get older, I ain't got the energy to even run a fever. So apparently the elderly don't hardly run fevers. Something to look forward to as you get older. I can't generate enough energy to get this thing going. What am I going to do? Because I got to break down carbohydrate polymers. I ate a Twinkie. I need to extract energy from it. I got to set it on fire somehow. What am I to do? Glad you asked. What we're going to do is we need a way of lowering the energy of activation. Ta-da! We have the way. And that is bringing in a catalyst. So a catalyst is a compound, molecule, whatever, stuff that lowers the energy of activation. That's cool. And generally, in fact, always for the catalyst, it's not being consumed in the reaction. It's actually just lowering the energy of activation. As it turns out, we have in our molecular bag of tricks proteins that are catalysts. How about we'll say it this way? Proteins that act as catalysts, and we give them, of course, the special name of enzymes. So enzymes are protein catalysts. Hey, that's going to work. Now, we mentioned, and just for completeness' sake, because this is being recorded, pretty much all the enzymes we'll be talking about from here on out are protein-based enzymes. They're amino acid polymers. There are RNA sequences, as we finish up on recording on Friday, RNA sequences that can fold up to actually generate catalytic activity, but pretty much all they do is break apart other RNAs. And yes, we have ribozymes, but I don't want to talk about them. All right, so we've got protein catalysts. And what they're going to do is lower the energy of activation. And so there we go. We've got now the enzyme catalyzed energy of activation. And if I'm lucky, the energy of activation has been lowered enough that I can carry it out at body temperature. Wow, that's going to rock. Now, rather than having to have 700 degrees to get this fire started, I can do it in the comfort of my own home. Now, interestingly, outside, they ain't a whole lot of energy on a day like today. Consequently, even with enzymes, plants, our little bug friends, they're having a pretty rough time trying to carry out any kind of catabolic reactions, not to mention anabolic reactions. Forget about it. So when things get cold, unless you can generate your own body heat, things are going to get really, really slow. Freezing jacks up everything in the world because ice crystallizes, and when it does so, it ruptures the cell membrane. Freezing is just a mess. But let's just say, okay, we warm up 30 degrees or 35 degrees, and we're right at just above freezing. So the cells are still intact. Proteins, cold doesn't really denature proteins terribly, at least permanently. It will slow them way down, their ability to do stuff. But if things warm up, the hydrogen bonds will kind of rearrange a little bit, and stuff will get rolling again. So cold's not nearly as bad as a whole lot of heat for life. But the problem is there just isn't enough thermodynamic energy to begin with out there to carry out these reactions. So, I can lower the energy of activation, but I can only lower it so much. All right. By lowering the energy of activation, we can carry out the reaction. Now, this works really well in terms of definitions for our catabolic type reactions, breaking stuff down. And my favorite example is where you're going to get your roommate and promise him that you're sorry that you electrocuted him, but you got a better experiment now. You borrow his car and you take it up to the top of a really tall hill, and particularly one that would have like a sharp drop, a cliff. Like, I don't know, we'd probably find one around Lake Decatur. And you say, I got a trick here. This is going to work just fine. It's a good example. And you bring the car up, and we got one of those parking lot bumpers. Oh, it's about this high. And you nudge his car right up to that parking lot bumper. Now you put the car in neutral, and you go to the back of it, and you start doing this. And your roommate's getting a little nervous. But it's okay. You're a scientist. You're good. And, with a parking lot bumper like that, man, you're not going to get that thing to come over. So that's kind of what is happening at a moderate temperature, that kind of energy. Now, if you say, let's swap that parking lot bumper out for just one of them little rubber guys that maybe you have in your garage, a little stop. So we'll roll over the bumper from there oh, to about there. Roll the car up to it, it stops, all's good. And now you go, and his car rolls off the hill, and you, sorry, okay. That is what we're doing by lowering the energy of activation. Now, an example of, a non-catalyzed reaction would be, we got the great big parking lot bumper, but instead of you just doing this, you get the entire football team, and they're all pushing on it, kablam! And so with enough energy, enough heat, you can get over the energy of activation. You get a bunch of big guys on there, man, they can push that thing off the cliff. They can get it over, a really high energy block. But with an enzyme catalyzed reaction, it don't take that much energy. Now, just as we're kind of thinking about these things, things that have a higher energy of activation, we would say, hey, that's a pretty stable molecule. Things that have a really low energy of activation, you're going to go, damn, that stuff falls apart really fast. That's like really delicate. And upstairs on the third floor, we got the -70 freezer. And I store stuff in the -70, which is really, really cold, colder than outside, that is labile. Things like adenosine triphosphate. That third phosphate, it falls off really easily. It decomposes. If you leave ATP out at room temperature for a couple hours, that third phosphate probably falls off because it has a really low energy of activation to break that bond. So things that are really stable, I like, because I don't have to be all that careful with them. Things that have a low energy of activation, they decompose very, very quickly. So we got a range of different kinds of bonds and molecules within the living system. Some stuff, phenomenally tough. Leave it at room temperature for years. Don't worry about it. Other stuff, man, you better buy it fresh. You better split it out, aliquot it, put little bits into a tube, stick them into a freezer so you don't have to freeze thaw it. I mean, it's like really a pain. There are a whole range of different kinds of molecules that I pretty much have to buy, every year, even though I've got, that much in a tube. Nah, it's dead. Let's not even bother trying it. So we've got a range of different kinds of molecules and a range of different kinds of activation energies, but we can lower them by using the catalyst. Now, that is for catabolic reactions. I like the analogy. I'm down with that. For anabolic reactions, it's a bit more complicated. Because now what we got to do is build stuff, and we have to add energy into the system. But here is another good sort of generalized analogy. Imagine that we're going to build something. We're going to start putting atoms together. And just for the sake of our diagram, we got molecule A, and we got molecule B. All right, really creative here. And remember we talked about making covalent bonds, we got to bring the atoms close enough together that the electrons can be zooming around each other. And we have, in fact, the Coulomb barrier, which I always think is very cool that I can use that term precisely. The repulsive nature of the electrons means that in order to get them close enough, you got to put a fair amount of energy into this stuff. So building bonds, man, is not trivial. I mean, in fact, we can know that the same energy that you get out of a bond, that's the energy you got to put into the bond. And so if wood generates a good amount of heat, it's going to take a fair amount of energy to make that chunk of wood. So we got a little bit of an issue here. We got to bring these molecules together. We have to overcome these repulsive forces. And I'm just looking at my clock there. All right, so this is a possible mechanism. We have an enzyme that looks like this. It binds the two parts, and it's got them in what we call the active site. So we got the two parts here. We got to have a way to slam them together with some force. How about this? Let's go ahead and have a whole set of negatively charged amino acids down here, and we'll put specifically a neutral amino acid. This is going to be either a serine or a threonine. And this is rather nerd detail, but we'll get into it in just a moment. Serines and threonines are near neutral, very weakly charged amino acids. But then around them, we're going to have a whole bunch of amino acids that are carrying negative charges. So they may be folding up into some sort of alpha helical structure. Regardless, we've got a lot of negative charges here. What if we take a phosphate and throw that on to the serine or the threonine? We'll just make it a serine just to keep the diagram simple. So now we're going to make a phosphorylation event with that massive negative charge. Remember, phosphates, negative charge. And what's going to happen here? We got a whole series of like charges, but now we've added a massive negative charge that's going to be pushing these guys away. And in a bit of blackboard animation, this one I wish I had like a living board, that would just go whoop, but the whole thing slams in, bam, with massive force, like slamming a car door. We got A right next to B, and they have formed a bond. Yay! It's a bit simplistic, but hell, it works. This actually is a standard way we can build molecules, and the number one thing is it brings in the notion of the phosphate. What is our number one phosphate donor, you might ask? It is, in fact, the ribonucleotide adenosine triphosphate, ATP. And so that old sort of diagram you have of ATP goes to ADP, what that means is the phosphate is being removed. We've got adenosine diphosphate. That third phosphate is right there. And this is, in fact, a critical reaction. When we add a phosphate to a serine or a threonine, and also tyrosine, but we'll get to those later, this is called a kinase reaction. The enzymes that catalyze the addition of a phosphate onto something is called a kinase. Kinases rule the world. Kinases are driving everything. Kinases are fundamental to cell biology. And what they're doing is transferring a phosphate from the triphosphate onto a molecule, generating the diphosphate. And a kinase reaction is often what's driving these kinds of anabolic reactions. This is, of course, somewhat oversimplification. We'll see as we go through this. A lot of times, we'll have, all right, we need some energy here. ATP goes to ADP. And what we mean is that phosphate got put onto something. What you can do is you can pop that phosphate off in what's known as a dephosphorylation event. There are enzymes called phosphatases that remove phosphates. And you can now take this protein and change its shape. Adding the phosphate, blam, it clenches up. Remove the phosphate, it opens back up. It works. So the phosphorylation, dephosphorylation is critical to a lot of things. That phosphate is being donated by ATP, and there we go. Now, as I say, as we go through this, there are a whole range of different biochemical reactions that we'll say, okay, ATP is being used, it goes to ADP. Sometimes it is, in fact, a covalent phosphorylation kinase event. Other times, we'll have a little pocket where ATP will dissociate, donate the phosphate, and kind of leave it hanging in there. And while it's not covalently linked, it's still imparting its massive negative charge, and so that works as well. But this actually is the way we are able to move, we're able to physically move, but you're also able to transfer energy around by moving phosphates around. And it works. That's all I got to say for today. And there you go. So I will catch up with you on the video for the next 2 lectures, and we'll see how that goes. So we're not doing lab this week. I'll let you know. We're good. That's all. Bye.